Okay, magandang buhay everyone. So now we will be having our new lesson, which is the another type of cell division, which is the meiosis. So meiosis. So meiosis is a process that contributes to the genetic variety. They do not make body cell; it makes the sperm cell and egg cell, also known as gametes. So remember, in mitosis, the cell involved is our somatic cell or body cell. In meiosis, the cell involved are our sperm cell and egg cell, which is we call the gametes or sex cells or reproductive cells. So meiosis also called as the reduction division because the starting cell they have 46 chromosomes and the ending cell will have 23 chromosomes. Okay, so human cell, remember, we have 46 chromosomes. That is the number of chromosomes in most of our body cell. But some cells they have 23 chromosomes for X, and that special cell are our gametes or the egg cell and sperm cell. So egg cell and sperm cell they have 23 chromosomes. So if the sperm cell have 23 chromosomes or the chromosome from your father is 23 and the egg cell from your mother contains 23 chromosomes, when we combine it, we can make 46 chromosomes that will allow a new fertilized egg, which also called the zygote, to develop into human. So before meiosis, remember, the cell undergo interphase, and then in interphase, um, the DNA is being replicated, right? Or all organelles are being replicated. So when we have 46 chromosomes duplicated, we still have, of course, 46 chromosomes because the sister chromatids are still attached to each other. Because the number of chromosomes are the number of centromere. Okay, so we have 46 chromosomes, but we have 92 chromatids because remember during interphase they are being replicated. So if we have 46 chromatids and then they are being replicated, we will have 92 chromatids, but we have 46 chromosomes because we have 46 centromere. Because centromere, it is the center of chromosome where two sister chromatids are being held together. So in meiosis, the first or the starting cell have 46 chromosomes, and then it will be divided into two, which is the meiosis 1. They have 23 chromosomes, and then after that, that 23 chromosome also be divided into two, which will undergo meiosis 2, and then after the meiosis or the actual or the whole process in meiosis, we will have four different cells. Okay, in mitosis we have two identical daughter cells or same cell. In meiosis we have four cells that is not same with the starting cell. Okay, so meiosis it have two type or two cell division. So we have the meiosis 1 and meiosis 2. So the name of the phases is also same with the mitosis. So the first phase or in meiosis 1, the first phase is the prophase 1. So prophase 1 are condensed in the nucleus and they are breaking down and the nucleus or are being so the nucleus is not visible or it disappear. So in prophase 1, this is where the crossing over happened. So when we crossing over happened, so it happened in the homologous chromosomes. So meaning when we say homo, meaning same. So the crossing over happens if the chromosome contains same genetic materials. For example, are that chromosome contain the eye color. If the if your mother have blue eyes and your father have brown eyes, those chromosome that have that gene specifically that gene, the eye color, will cross over. After that, the crossing over, we will have the recombinant chromatins. Remember, the crossing over is the exchange um between it is exchange in genetic material that explain why some siblings, even though they have same parents, they are not really identical to each other. So for example, the firstborn, um, she have brown eyes, and the secondborn, she have blue eyes, because the mother and the father, so they have two different types of eye color. So next bit in prophase 1 is the metaphase 1. So metaphase 1 is the same with metaphase in the mitosis, but in metaphase 1 or in meiosis 1, the chromosomes are aligned in pairs. Pairs meaning, remember in metaphase, it is single 5, but in metaphase 1, it is aligned in pairs. So random assortment occurs, meaning the chromosome per line randomly. Next, we have anaphase 1. So anaphase 1, A4, away. So the paired chromosomes are being separated and moved in the opposite ends of the cell. So this ensures that each cell contains half of the number of chromosome or haploid. When you say haploid, it is the half of the number of the chromosome. The next phase we have telophase 1. So telophase 1, the two nuclei form around each set of chromosome. And then after that we have, of course, the cytokinesis, which we have now two cells. And then now the cell is done with the meiosis 1. And then the next division is meiosis 2. And in meiosis 2, they have also the same stages. So prophase 2 it is just the same with the prophase in mitosis. So in prophase 2, the chromatids are getting condensed and thickened, and then the nuclear membrane disappear. And the spindle fibers will attach to the centromere because they are able to attach to the centromere because the nuclear membrane again disappear. And then in metaphase 2, the chromosomes are aligned in single file in the center or in the middle. So this occurs that to ensure the sister chromatid separates in the next stages. So the next stage is anaphase 2. So in anaphase 2, of course, the sister chromatids are being separated and move in the opposite end of the cell or the opposite post in the of the cell. So this ensures that the daughter cell remain haploid, so meaning it have the half number of chromosome, which is the 23. This means that they have half of the genetic material from the original parent cell. So meaning, so that daughter cell they have um, half of the genetic material from your father and your mother. So next, of course, we have telophase 2. So four new nuclei form around each set of chromosomes. So the cytoplasm split and four haploid daughter cell form, that is in the form of cytokinesis. Cytokinesis. So this is the whole process in our meiosis. So remember the difference between mitosis and meiosis. In mitosis, after meiosis, we will have two identical daughter cells. In meiosis, we will have four different cells. Because of the independent assortment and the crossing over, you are going to have a variety. For example, four sperm cells produce each time, and they are all different from each other because they are different from the starting cell. Because remember, the crossing over happened in prophase 1. And then in mitosis, the cell involved is it occurs in our somatic cell or body cell. In meiosis, it occurs in our gametes or sex cells or reproductive cell, which are the sperm cell for male and egg cell for female. Okay, so that's it. And i-discuss naman natin din to pag nagkaroon na tayong face-to-face. Mas madidiscuss natin to. So, ayun. Thank you and God bless everyone.